I'm Dr. Feng-Ming Tian from National Taiwan University, and I'm an attending physician in our department for around 10 years. And my clinical service includes treating the acute leukemia, lymphoma, and myeloma patients. But my research interest is focusing on the acute myeloid leukemia. And I'm focusing on the risk stratification of AML patients by the commutations. And now I'm digging deeper into the mechanism of AML, and I'm now working on the bioinflammatory mechanism of AML patients. Today, it's my honor to be selected as an oral abstract in this SOHO Asia meeting, and my topic is focusing on the immune landscape and treatment response of azacitidine in both the high-risk MDS patient and acute myeloid leukemia patients. And I think previous studies has more focusing on the tumor cell, the myeloblast itself, to see whether there are some tumor characteristics that could predict AZA response. But actually, there are no convincible markers that could predict the response. Since some of the research has found the immune signal pathways, such as the interferon gamma signaling, may be related to the azacitidine response. So we use our bone marrow samples to profile the immune landscape in these patients. So we use both the RNA-seq analysis, and we also use the single-cell analysis by CyTOF, the mass cytometry method, to profile both the methylation and the immune status of our patients. And we find out that AML has a unique immune cell compartment, such as the increased CD8 T cell subsets among the CD3 population. And interesting, some of the CD8 positive cells, they are within the CD8 positive T cell, more CD7 positive and CD60 positive and CD57 positive cells are also weakness. That means that the CD8 T cells are more senescent and more NK-like in the MDS and AML patients. So this means that the immune microenvironment actually is more detrimental in these patients. So we next want to find whether this immune context predicts AZA response. So we have profiled the RNA-seq between the responders and non-responders, and we have found that more enriched immune pathways such as the interferon signaling and toll-like receptor signaling are associated with better response. So this means that if we want the AZA to have a response, the immune system must be reprogrammable. The immune system must not be too weak. So that if the immune system is too weak, then AZA will not work for these patients. And we also find selective markers such as the TLR4 and HLDR expression. These markers, higher expression of these markers also correlate with a better response. So this study highlights that apart from studying the myeloblasts, some of the clues are in the immune systems, and they really work to influence the responsiveness to AZA treatment. I think since the MDS and AML are very heterogeneous disease, so we are now trying to dissecting the heterogeneity within this group. So far we have done this by study a lot of commutation pattern and transcriptome signatures that could separate AML with the same gene background. But in the future, I think a more detailed analysis, such as the methylation landscape, the chromatin accessibility landscape, should be analyzed within each AML subcategory. So this will help us to pave a way toward a more risk-direct treatment for these patients. I think this is indeed a very high-quality level meeting because from the speakers, they are very well known, and I think their presentation are very fantastic studies and very new concepts. And we also have a chance to have a talk with the speakers last evening in the gala dinner. So I think this meeting is very, very good, and I sincerely hope that SOHO Asia will be held again. Of course, I think the SOHO in Houston is a must for all hematologists.